Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is A Long Read versus Short Read Platform Comparison, Best NGS Approaches for Microbiome Studies, and our sponsor is Loop Genomics. Our panelist today is Nick Greenfield, founder and CEO of OneCodex. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you don't see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentation has concluded. Mr. Greenfield, please go ahead with your screen share.
Great. Thanks so much, Christy. So I'm just starting to share my screen here. Hopefully it's coming through. Perfect. So today I'm going to be presenting a bit of data on long-read LoopSeq 16S data compared to short-read V3, V4 Illumina sequencing data, as well as whole genome sequencing or shotgun sequencing short-read data. And I'm going to talk about these three different data types in the context of microbiome studies and some benchmarking that we've done of these different data types across known mixture community standards and talk about the accuracy of these different techniques and the implications for thinking about and designing your microbiome studies. So as a brief agenda, what we're going to cover here today is first, I'm going to provide a little bit of background on OneCodex and an introduction, and then I'm going to actually demo our system. So we've built a platform for analyzing microbial next-generation sequencing data, and I'll provide a demo of that specifically in the context of how to analyze LoopSeq 16S data. So we'll walk through an example. We'll actually drag and drop some data into the website, upload it, get a feel for how to interactively explore that data and probe it a bit further. And then we'll take a look at long read versus short read sequencing data and a bunch of analysis that I and the team here has prepared, comparing, again, Illumina short read V3, V4 sequencing data, LoopSeq sequencing data for 16S, and shotgun sequencing data. And then we'll conclude and take time to wrap up. Great. Are people having trouble with the audio?
No, Nick, the audio is fine. We'll address audio issues on our end. Don't worry about it.
Perfect. I just wanted to make sure everyone could hear me. I saw one message from someone coming in saying, "Is the audio working?" And I was hoping folks were catching this. Okay, great. So, by way of brief introduction and background, so OneCodex, we're a San Francisco-based microbial genomics company. We're entirely a dry lab operation. So we're really a software engineering organization with microbiology expertise. And we've built a data analysis platform called OneCodex for analyzing various microbial next-gen sequencing data sets, whether those are microbiome samples, stool, saliva, skin swabs, et cetera, bacterial isolates, or environmental samples. This is all a cloud-based system. There's a little screenshot on the bottom left we'll walk through and kind of show that system live in a few minutes. And at the core of what we do is we provide a bunch of different types of analysis on our platform. So we have a curated database of over 80,000 whole microbial genomes, and we compare shotgun metagenomic data or whole genome sequencing data against that database and try to provide best-in-class answer to the question of who's there, what are the bugs that are present, as well as a little bit of what are they doing. So we do some work around functional gene panels, other types of analyses. And today we'll be focused a little bit on our database for what we call our targeted loci database, which is our database for 16S, 18S, ITS, and other common marker gene sequencing schemes, so for use in targeted sequencing such as 16S analyses. And our platform's used by folks. This is kind of a snapshot of academic and public sector end users, but we've got over 5,000 users on the platform across a bunch of different institutions, including academic institutions, folks in public health, and elsewhere. So kind of widely used and tested for microbiome research in both the academic sector and as well as commercially. So today we're going to be talking a little bit about, I think most on the line are probably familiar with short read, V3, V4, or other variable regions 16S sequencing on Illumina's platform. What we're going to be talking about today and introducing is specifically looking at LoopSeq data, which is a type of synthetic long read data that can be produced with a kit from Loop Genomics, pictured up here in the top right, and that focuses on, that has some unique biochemistry where basically they attach unique barcodes to the individual molecules, they amplify up the molecules, they have some chemistry for then distributing those barcodes randomly across the individual molecules, and then they do library prep and sequence. And so each of these individual molecules is uniquely barcoded, and then they have some computational biology and assembly at the other end to generate basically synthetic long read data where there's a lot of coverage across the individual molecule, and so you get a very accurate consensus picture of the input molecule. And in this case, specifically, the kit that we're going to be talking about, and the use case we're going to be talking about, is full-length 16S sequencing. So rather than sequencing one variable region, sequencing across what you can think of as V1 through V9, across the 9 variable regions of the 16S gene, and then generating a consensus sequence that has a barcode tied all the way back to the individual molecule. And I'm sure Tuval, who's on the line and is the CEO of Loop Genomics, can answer some more detailed questions about this, but that's the general process. And that gets us synthetic long read data that is a consensus sequence. We'll talk about what the properties of that consensus sequence are, and covers all the variable regions in the 16S gene, and we'll talk about some of the benefits of that from an informatic perspective and from the perspective of thinking about how that implicates and microbiome sequencing and what that provides downstream. So with that little bit of background on LoopSeq, the LoopSeq 16S kit and their technology, a little bit of background about OneCodex and who we are, what I'd love to do now is actually step out of this PowerPoint presentation and step over to a live demo of our platform. And I'll focus on just providing a very cursory tour, but specifically showing how you can get started and analyze LoopSeq data within our platform and some of the properties of that LoopSeq data. And we'll do a little bit of comparison, and then we'll turn back to the presentation and go through some more detailed stats and graphs and figures that we've put together comparing the LoopSeq data and the short read data. So what I'm doing here is I've just gone to our website, OneCodex.com, and I'm going to click Login, and I've prepared a demo account for this webinar, and so I'm just entering my email and a demo account. If I didn't have an account, I could click here to create a new one. We won't save this password. And so when I'm logging in here for the first time, if you logged in, this would normally be blank. There'd be some getting started instructions. Here, I've pre-populated this account with 3 samples. And these samples are V3, V4 short read 16S sequencing data from Illumina, shotgun sequencing data, also on an Illumina platform, and LoopSeq 16S data, which was sequenced on an Illumina platform, but generates these synthetic long reads that span all of the variable regions within 16S. And one thing you may notice is there are some, all the basic functionality you'd expect in the system around metadata. So we can see there's some structured metadata on these samples. We can kind of store a bunch of metadata alongside of our samples. Tagging, we can filter. In this case, it's not very interesting because I only have 3 samples. But we can kind of browse this data in a bunch of different ways. And getting started and uploading the data is really just as simple as dragging and dropping a file into this upload or import pane. And I have a little file, just in case the internet wasn't so fast, and I'll upload a larger file here as well. But so this is some demonstration data. This is LoopSeq 16S data. And we see if we navigate back here, here are the two files that I just uploaded. They'll take a minute or two to process. We can look at some of this other data in the meantime, and then we'll come back to them. But getting started is kind of as simple as dragging and dropping data into this upload pane. We also have command line tools and APIs for pushing data programmatically to the system. So this file is actually already processed. Here, this is being analyzed against our OneCodex database. Our system's actually automatically detected that this is LoopSeq 16S data, and it's also performing this special analysis using our marker gene database or targeted loci database optimized for LoopSeq data specifically. So that'll take a minute to process, and we can come back to it. But that's kind of the basic interface for sample management, for getting data into the platform. What we'll turn to now is looking actually at one of these samples. And so this is actually a specific analysis for the ATCC MSA 1003 positive control, which is what this sample is. And what I really want to look at for the purposes of this demo is just kind of giving a sense to folks of what a standard analysis on our platform looks like. And this is kind of our who's there taxonomic classification snapshot, in this case with the targeted loci database for 16S. And there's a couple high-level things. So we present a summary panel. What are the high, medium, and low abundance organisms in this sample? We present some sort of interactive graphics around the composition of the sample. And we can look at this through the lens of being only interested in the species that are present, or the genera that are present, looking at a higher order taxonomically, such as the family, order, or class, or phylum. We can also kind of explore this interactively in a kind of sunburst or chronoplot style. So a bunch of options for kind of interactively exploring this data. For short read 16S data, we generally recommend looking at things at the genus level. For the LoopSeq data, we can actually get down to the species level, which is something we'll talk about. And the same applies to shotgun data. And then there's information about how the reads map taxonomically across a phylogenetic tree. Again, kind of focus here is on ease of use, interactivity. You can export these images if you need to. You can export all of the data that we're looking at here programmatically in a variety of formats as well, and tabular information. So this sample, if we kind of eyeball it at the genus level, we see there's 3 high abundance organisms, then there's a cluster of kind of modest abundance organisms, and on and down. And that actually is reflective of what we know about this particular sample, which is some gDNA in a known mixture from ATCC that has 20 organisms and is staggered. And so we see that composition come through here, and we actually have a specific kind of scorecard analysis for the ATCC materials as well, and we can see what are the different organisms that are present, what percentages do we expect versus what did we observe, et cetera. And you can see these are staggered. So some of the things are expected at relatively high abundance, and some are expected at very low abundances. Those were the true positives, sorry. High expected and lower expected abundances. And now if we turn back to the LoopSeq demo data, we can see here, this is the sample that we uploaded, the larger sample. It's 51,000 reads. These are synthetic long reads, so they're going to average, probably about 1,400 bases, given that we're targeting the 16S gene and all of its variable regions, so that'd be approximately the length that you'd expect. We're classifying the vast majority of these reads, and again, we see this is the same input material. We see, in this case, 4 kind of high abundance organisms, and then the staggering on down. We can look at this data from at kind of the species level because the longer reads give us a little bit more insight and greater specificity in classifying. But you can see same input material, very similar mix, tabular data, et cetera. And we can actually compare this, grab the LoopSeq demo data, compare it to the short read data, compare it to, this is the shotgun sequencing data. I want to normalize these and kind of look at, compositionally what's there. Well, these look pretty similar. If we look at them at the species level, again, they look pretty similar. I think the thing to observe is we just drag the LoopSeq data and put it in the middle. You can see the LoopSeq data and the shotgun data look very close. The 16S data is a little bit more divergent. And we'll talk about some of the statistics of that in a moment and kind of dive in and explore that a little bit further back in the presentation. So those are some of the basic features, just to give folks a sense for context in how you can start analyzing this data type in our platform and how you can analyze other microbiome data in our platform. There's a bunch of things we've not covered that we can take questions on at the end around comparative analysis and built-in notebooks to the platform and some of the specific features that are designed for these known composition positive controls from ATCC, of which there are a great many, and we're specifically kind of looking at a bunch of data from this mixture of 20 bacteria here. Great. So I'm going to switch gears, use my 10-minute demo, and move back to the slides and dive in a little bit to the analysis that we've done of this long and short read sequencing data. And to tee it up, I think these are the three data types that we're really talking about. So we have short read 16S data. I think this is by far the vast majority of sequencing that's been done of microbiome specimens has been short read 16S data. V3, V4 is a very common variable region to target. You typically do this on a MiSeq with 2x150 sequencing. You also see folks looking at other targets like V1, V2, or looking at just one variable region and trying to get shorter reads. But so I think short read 16S data is something that we all kind of know in the microbiome field, and it has some advantages in that it's well, there's a lot of literature, so it's somewhat cheap, it's somewhat ubiquitous, but it has a lot of kind of data disadvantages as well in terms of the specificity of the results that are generated, some abundance issues, et cetera. We'll dive into those. And we're really comparing short read 16S data with long read 16S or full length 16S data, in this case from the LoopSeq 16S kit. And then we also want to look at that in the context of short read whole genome sequencing or shotgun sequencing data that you might see coming out of a Nextera library prep or similar. And I think the question is, how do we want to look at these different data types? How should we compare these methods? Someone comes to us and says, I'm planning a project, I want to sequence 500 stool samples. What should my technique be? What I have the following questions. Should I be using short read 16S? Should I be using LoopSeq? Should I be using shotgun sequencing? And I think the question, what we want to do is provide a little bit of ground truth based on known mixtures to be able to look at, what do these different methods provide in terms of the picture of the composition or the community profile. So how accurate are they at kind of recapitulating what we believe to be there in terms of the composition of organisms that are present? How specific do they do that? So with short read 16S data, it's typical that many of those variable regions for some species are non-specific. So, you'll sequence the V3, V4 region, you'll have a large number of reads, regardless of if you're using an OTU clustering approach or a reference-based approach, but you'll have a large number of reads that you simply don't know what they are at the species level. You may know that they are bacillus species, or you may know that they're Bacteroides, or Bifidobacterium even, but you don't know that it's Bifidobacterium anomalus, for instance, because that variable region is conserved across those different Bifidobacterium, and so we don't have the information content to make an inference there. And then I think the final thing that is important in thinking about kind of which of these methods to choose from is looking at actually the sequence level accuracy, so the base level accuracy of the sequencing data that results, and understanding the implications of that for our microbiome analyses. And how we're going to do this, and what we've done here in terms of presenting and teeing up some data, is we've looked at known mixture microbial communities, and specifically we've looked at as I said, the MSA 1003 20 organism staggered mixture from ATCC. And we have some analysis built in we have a partnership with ATCC and some analysis built into our platform that's kind of a easy to use scorecard customized for that. And then we've also looked at another control from Zymo called the Zymo Biomics microbial community. That's a 10 organism mixture, 8 bacteria, 2 eukaryotic microorganisms that we can also use. And we'll show data from both of these known community standards and talk about kind of the implications of what those mean across these 3 different data types. And we only have 16S data for the second one. For the ATCC, we have the shotgun data, the LoopSeq 16S, and the short read 16S. For the Zymo, we only have the 16S data. So this is what that standardized scorecard analysis looks like. This data set specifically is the LoopSeq 16S data set that we're going to look at. I should note that this is probably the highest score we have ever seen on this scorecard. So that the LoopSeq 16S data performs very, very well in terms of kind of describing this mixture and recapitulating the input here. And I think one thing, it says all 20 organisms are present. It does a very nice job in terms of thinking about the relative abundance. But I think perhaps the most impressive thing is that there are no false positives in that data. We'll dive into that in a little bit. So it provides a really nice snapshot of what's there, and it doesn't mislead us and have us chasing our tails or going down paths exploring organisms and relationships that might not really be supported by the data. So as I said, we're going to look across 3 different metrics, or think about 3 different these data sets from 3 different lenses. First, we're going to talk about composition and how well each of these techniques reproduce the theoretical composition of these different standards. And the score that we like to use here at OneCodex for looking at this is something called the Aitchison score. This is 1 minus the Aitchison distance. And the formula is up on the top right, but basically this is a compositional distance metric. So it's designed, I believe the origin is actually geology or geochemistry, but it's designed specifically for thinking about, what are the differences between 2 things that are mixtures where we only have relative abundance information and everything sums to 1, which is what, sequencing data currently gives us. It doesn't give us absolute abundances, cell counts, things like this. It rather says, this community member has gone from 10% to 30%, and that's all that we know. We don't know if it's tripled or if other things are depleted. So this is a nice adjusted metric for looking at the overall distance between a theoretical composition and an observed one. And 1 is a perfect score, of course. So shotgun sequencing data does very well on this ATCC standard, something like 96%. Which you would expect because it's effectively random sequencing across all the microbial, across all the DNA in those samples. In this case, all of which is microbial. But and then short read 16S here at the bottom doesn't do so well, a little bit under 80%. And LoopSeq performs, I think, very impressively here, given that we're not looking at all the genomes, we're specifically looking at the 16S gene only in the kind of high 80s. And we're going to focus really on this comparison because I think these are, in many ways, the comparable technologies when you think about some of your kind of experimental scope and cost and other kind of practical considerations. So that's a little bit on composition and the lens which we're going to use to look at these 3 sequencing libraries and the data that are produced. And here is a chart of the theoretical abundances of the different organisms that are present in that ATCC mixture. And as I mentioned, there's 20 organisms. We see that they're staggered at 4 different levels across, I think, let me do my math really quickly, 3 logs, 4 logs, and 5 in each of these tiers. And the whole genome or shotgun sequencing data looks pretty good, right? We clearly observe these 4 tiers, and in general they're really quite even. The LoopSeq data, the 16S data, also looks very good. So again, we still have these 4 clear tiers, fairly even distributions across these individual genomes, When we look at short read 16S, this is where things start to look a little bit messier, right? And you start to have kind of a bleeding effect between some of these individual tiers. Like, it's not immediately clear if you were just eyeballing the rightmost chart and you saw this pink bar down here, was that supposed to be at the lowest input amount or the second lowest input amount? Similarly, this blue bar is significantly below a bunch of the things that are supposed to be at the lowest input amount. And here I'm overlaying kind of what's the expected, and you can kind of see this visually, where some things are being amplified up, and we're seeing more of them than we would expect. We're seeing gaps, again, across, and then we see things bleeding in, and it's kind of unclear what the actual abundances are. So compositionally, we're losing a lot of the picture here about what that community actually looks like and the relative abundances of these organisms to one another with the short read 16S data. And here's both the ATCC control on the top as well as the Zymo known community control on the bottom. And this is kind of a picture of how specifically do we classify the individual input reads. And with the LoopSeq data, the vast majority of the data is specific to the species level. And there are some artifacts here of the database and exactly what organisms are present. And I believe it's the case that kind of one of the organisms present in the ATCC standard, of which there are 20 organisms, is just not really specific in terms of its 16S gene. And so we basically see 19 of the 20 organisms. We can make species-specific calls, so that's approximately 95% of the LoopSeq synthetic long reads are mapping down to the species level, and that remaining 5%, we're not making the specific calls, and that's probably due to the underlying biology. In contrast, short read data, in the ATCC case, about half of it we can make kind of species-specific calls. In the Zymo case, it's like 30%. So in general, we're seeing far fewer species calls. We'll talk in a moment about false positives. Some of these species calls are erroneous, and they're due to sequencing error, looking like known real variants. And we have a lot of data that's just fairly nonspecific. So we have a lot of reads that map to the genus level or that map to the order or family. These aren't quite in order. Class, etc. And so each of these reads, doesn't tell us as much. Our ability to make inferences about our sample, about the composition of a complex sample like stool, is kind of greatly reduced when instead of a bag of species, which is what the LoopSeq data gives us, we're kind of getting a bag of, sometimes we know it's a species, and sometimes that's because of the variable region. Other times we don't know if it's a species, and we just have information about the family, and we have a kind of messy, ragged, or staggered hierarchy of information about what we're observing and which bugs might truly be present in these samples. And this specificity is effectively a function of read length. So the LoopSeq data is here in blue. The ATCC community is on the left, the Zymo community is on the right. And you can see across both of these, the kind of median of this distribution is at about, about 1,500 base pairs or 1,450 base pairs or something like that here. And that's what you'd expect because that's the full length of the 16S gene in kind of the average case. And then we've got, a number of reads, synthetic long reads that are at lower read lengths, and that's cases where there wasn't enough coverage over the molecule to assemble a complete picture of that molecule, and so maybe it's broken into two pieces, or maybe there's just coverage over one part of it. In contrast to the short read data, and this is MySeq 2x150 data, right? So this is kind of as long as you can get on Illumina. It's all centered at exactly 300, which is what you'd expect. 2x300. Excuse me. And yeah, these are the individual. So this is 2x300, and these are of the individual forward and reverse. They're not the paired. It would be at 600 if they were joined, depending on the overlap. And here we're looking a little bit at sequence accuracy, which is the other major driver of of thinking about the species composition and both being able to make species calls, but critically not making species miscalls, resulting in false positives. And you can see that, on the Zymo standard, the LoopSeq data is, there were like a very large number of nines. So with SigFigs, we're rounding this to 100% accurate. Is the median identity, base level identity, and it's 99.9% on the ATCC control. And in comparison, we see 99.7%, which is fairly standard, what you'd expect from the short read v3, v4 sequencing data. And for those who are kind of less familiar with some of the nuances of 16S data analysis, you might say 99.7%. That sounds really accurate, and it is. But the challenge is that the 16S gene is so conserved that those errors often look like actual variants. And so a base mismatch at one position in the 16S gene might, lead you to say that, your Bacteroides ovatus is Bacteroides, something else. And so you might make a species miscall on the basis of those, that 0.3% error rate. You might simply just have a base mismatch that means you can't map to the reference with the same confidence, et cetera. So that decreases both the specificity of the results, and it also is a source of a lot of the mismapping problems for 16S. And this applies to both kind of reference-based paradigms as well as clustering. When you're clustering, your clusters just end up being a little bit bigger, a little bit less well-defined. When you then call them based on the centroids or depending on the method that you're using, you have these same types of problems. So, I think the ultimate implication of this is, this is that interactive pie chart that I was just taking a screenshot of the LoopSeq 16S data. Here, this is actually the Zymo community, which, as I mentioned, has exactly 8 bacteria, and we see exactly 8 bacteria, and notably, nothing else. So we have no false positives, and that I think is, perhaps the most useful thing about this is the extremely high confidence one can have in these results, even at fairly deep sequencing, rather than seeing a tail of data where sequencing errors contributing to possibly erroneous signal. In contrast, this is the v3, v4 16S data for the Zymo kit. We see the same 8 or so organisms at the top, but we see this tail of false positives. And I've actually truncated this. There's 23 in this list. It flows off the slide. And so, we have this just tremendous difference where we're both the distribution here. This is an even mixture, right? So the distribution is a lot more even than what we would expect. So the compositional picture is much better with the LoopSeq 16S data versus this, which is highly uneven and skewed towards, B subtle as being 14%. You'd expect it to be quite a bit lower. And we have this tail problem of tons of false positives in the short read v3, v4 16S sequencing data. And LoopSeq in this case addresses both of those. So, we've walked through kind of looking at short read 16S, v3, v4 sequencing, short read shotgun or whole genome sequencing, and LoopSeq 16S sequencing across the ATCC microbiome standards as well as the Zymo biomics microbial controls. And these are both known community mixtures, and we've kind of used those to benchmark and understand, how accurately do these various technologies recapitulate what we observe for the purposes of thinking about, approximating and measuring a stool sample or a saliva sample or some other human microbiome or environmental metagenomic sample. And, I think the things that we can kind of take away from this is that the LoopSeq data, provides a compositional or community profiling picture that is in many ways comparable to shotgun sequencing. Obviously, it has a bunch of benefits around price relative to shotgun sequencing, and as well as kind of on the laboratory side around ease of use and workflow. But if we're trying to answer the question who's there and get a snapshot of the community or a taxonomic profile of a sample, the LoopSeq 16S data provides a really nice picture, and is a great tool to have in the toolkit. If we care a lot about individual sequences and we don't want to be kind of misled or led astray, the LoopSeq data has, by its nature of being synthetic long reads and everything being consensus sequence, we're effectively squaring away the errors in the Illumina instrument and getting a very, very, very accurate picture of the individual underlying sequence. I think on that ATCC control where we saw 99.9%, it's possible that that 0.1% is actually, kind of error in our references rather than error in the LoopSeq data. That's how accurate it is. And, the final piece is that LoopSeq gives us species level calls and the ability to discern what are the individual organisms that are present far more specifically than we could do with short read data, short read 16S data, where, the typical recommendation that we and others provide would be that you'd want to look at short read V3, V4, 16S data at the genus or family level. With the LoopSeq data, you can really dive all the way down to the species level, and I think that makes a bunch of follow-on questions much easier to ask. It provides a lot more confidence about what's truly there. It provides better leads for your kind of follow-on questions in your microbiome experiments. The downsides here, obviously, you don't get functional profiling like you might out of shotgun sequencing data. The tooling is a little bit less mature because this is effectively kind of a novel data type, and you have a little bit greater sequencing depth requirements. Though you can do LoopSeq sequencing on kind of the cheaper per base instruments from Illumina. So you can, these workflows are compatible with putting it on a NextSeq or a HiSeq rather than doing it on a MiSeq or a MiniSeq where your per base cost is a little bit higher. So that's kind of our synopsis of looking at these three different data types across the ATCC and Zymo known community controls. And with that, I'd love to kind of take a step back and take questions for myself or for Tuval about the LoopSeq kits.
Thank you, Mr. Greenfield. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. And joining us for the Q&A today is Dr. Tuval Ben-Yehezkel, CEO of Loop Genomics. So we'll now start the Q&A. Our first question, Nick, can you explain a little bit more why the sequencing accuracy is improved with LoopSeq? And Tuval, if you'd like to jump in, please feel free to do so.
Yeah, sure. Actually, I think I have a slide explaining exactly that. Christy, is it possible to bring up my slides?
Yes, absolutely.
Great, thank you. Okay, so that's a very good question, and it's an important question because the underlying premise here is that we're using a short read sequencer from Illumina. So the question is, how are you able to get an error rate that's in order of magnitude lower than an Illumina sequencer if you're actually using an Illumina sequencer? So the answer is explained in this scheme here, where it shows that starting from the barcoding reaction, we have this barcode that's distributed intramolecularly to all the different parts of the molecule. And the end result in our prep is that for each UMI, for each barcode, we have generated a sort of cloud of short reads that collectively span the entire length of the 16S molecule. So typically for each position in the long read, we have multiple short reads that cover it. So errors in sequencing typically arise from polymerases, from just the inherent error rate of polymerases, and those are inserted during PCR. So if there happens to be a randomly introduced error in one of the short reads, as typically happens, we, in almost all cases, have other short reads that cover the exact same position in the long read that do not have that randomly introduced error. So computationally, the way that we assemble these molecules is through de novo construction. So that computational de novo construction of that molecule inherently filters out those errors through the process of consensus error correction. At each position, the identity of that base pair would be generated by the consensus from all the short reads for that position. And that, as it turns out, filters out the vast majority of errors in the long reads. We did this analysis on the Zymo synthetic community and on the ATCC synthetic community, and the error rate for the Illumina sequencing, short reads V3, V4 sequencing was approximately 1,600 errors per million base pairs, whereas the loop sequencing was about 40-fold lower at 40 errors per million base pairs. So that's kind of the high-level explanation of how our error rate is approximately an order of magnitude lower than the Illumina sequencer, despite the fact that we're using an Illumina sequencer. I hope that addresses the question.
And Nick, is there anything you'd like to add to that?
No, I think Tuval provided a great synopsis.
Okay, great. Nick, can you explain a little bit further how the wet lab part of LoopSeq works?
That's a question I think I'll also kick right over to Tuval.
Okay, great.
Okay, thanks. Yeah, for that I think I'll need my slides again. Okay. Right. So again, starting from our sample prep. So our sample prep starts with a barcoding reaction that specifically assigns UMIs, or unique molecular identifiers, also known as barcodes, to specific molecules. So each 16S molecule in the sample is assigned with a unique molecular identifier. That's a barcode written in DNA that's unique for that molecule. And then really the unique part of our chemistry is in the enzymes. It's kind of an enzymatic system that we've developed that enables us to, it's essentially a copy-paste operation in DNA that enables us to take this barcode sequence and copy and paste it into multiple positions within the same molecule, but not to neighboring molecules. So it's an intramolecular barcode distribution step. And that chemistry, this intramolecular chemistry, is really the chemistry that enables us to do synthetic long-read sequencing without the need to separate out molecules and physically separate them into different reaction chambers, as was traditionally used with earlier synthetic long-read sequencing technologies like those from Molecular or 10x Genomics that require that you either separate molecules, you do limiting dilution and you separate molecules into different reaction chambers, whether they are 384 well plates or water in oil droplets. But the idea is the same. You need to do limiting dilution and physically separate into reaction chambers that have different UMIs. In our case, since our chemistry is intramolecular, we don't have to physically separate the molecules, and the chemistry can be performed in a one-part reaction with all the molecules together in solution. You can think about this chemistry as sort of similar to transposases, where they randomly cut and paste a known sequence. But in contrast to traditional transposases that work intermolecularly, this chemistry works intramolecularly. So the barcode is copied and pasted within the same molecule into multiple positions. And after that is performed, we go through what is essentially a modified version of the traditional short-read prep, where these long molecules that have the barcodes inserted at different positions are chopped down and fragmented and made into a short-read library that can be sequenced on any Illumina sequencer. Now, the structure, the short reads are shown here on the bottom, and the structure of these short reads is always the same. The read always starts with the UMI, with a barcode, and then is followed by the actual sequence of the 16S, which was adjacent to where that barcode was inserted. So if this barcode was inserted into here, then the short read that will result from it will have the barcode at the beginning of R1, and then the actual sequence where the barcode was inserted. So collectively, when you take all the short reads that share the same barcode, you're then able to do computationally de novo reconstruction of that molecule into the full-length 16S molecule. And that is actually what our pipeline does. So when you purchase our kit, you get free access to our computational pipeline, which basically does this de novo reconstruction and generates FASTQ files with the full-length 16S data. Yeah, and I hope that addresses the question about how our chemistry is able to generate long-read sequencing from short-read sequencing data.
Okay, great. This question is, can you do whole genome sequencing LoopSeq on microbial communities? And if so, do things like Athena or Minerva work for LoopSeq?
So the answer to the first question, can we do shotgun metagenomics with LoopSeq? The answer to that question is yes, and we're actually very close to launching. Later this month, we will be launching a kit for single molecule long read sequencing of for metagenomics, and that will probably be subject to for another GenomeWeb webinar a few months from today. And regarding the second part of the question, I'm not sure that I'm familiar with was that Minerva? Maybe Nick, are you familiar with that? I'm not sure that I've.
Yeah, I think so. So I'm actually not familiar with Minerva offhand. Athena, I think, is a hybrid assembler out of the Bot Lab at Stanford. So we haven't taken a look at this question yet, but I think conceptually, a lot of actually what Tuval's assembly does is probably similar to the outputs of those kind of like other synthetic long read assemblers. In terms of providing, kind of coverage across those molecules that's consensus and high accuracy. But yeah, I don't know. We haven't played around with Athena specifically on LoopSeq data.
Okay. How do the results of LoopSeq compare with those of other virtual long-read methods?
So we're not familiar with other synthetic long-read methods that can provide full-length 16S amplicon data. I think that other synthetic long-read methods can provide data that's more similar to shotgun metagenomics or whole genome sequencing. So I'm not sure that there are other synthetic long-read methods that can do full-length 16S sequencing, but there are other long-read sequencing methods that can do 16S sequencing. For example, those from PacBio or Oxford Nanopore. We really haven't. This wasn't really the subject of the webinar here. We're focused on comparing it to short-read 16S sequencing, but we have done extensive studies on comparing to long-read sequencing. We actually have a publication that's in kind of final stages of the publication comparing LoopSeq 16S sequencing to PacBio sequencing data, and I can kind of give you a sneak preview to that. And well, the similarities between LoopSeq full-length 16S sequencing and, for example, PacBio data and Oxford Nanopore data is that they both provide full-length 16S sequencing. The two, maybe three major differences are, one is, as Nick mentioned, the sequence accuracy. So kind of referring back to Nick's slides, he showed the sequence accuracy between loop sequencing and Illumina sequencing and how it results in much less false positive identification. So that problem is actually even more severe with Oxford Nanopore and PacBio data, because generally their error rate is even lower than Illumina sequencing. So one major difference is the higher, the lower error rate and higher accuracy of Loop's long reads compared to Oxford and PacBio, resulting in much less false positive identifications. The other obvious advantage is cost. Since we're using an Illumina sequencer, the cost per base is lower. So compared to, for example, PacBio sequencing, our cost per base for long read sequencing is approximately 3 to 4-fold lower than PacBio. And then finally, maybe the last thing to mention is the UMI-based technology that does a lot of the corrections for abundances. So generally speaking, other long-read sequencing technologies like Oxford Nanopore don't employ UMI-based technology for doing for correcting for abundances. And just to kind of show you how critical this is. So this is what we call the PCR bias plot. So what you're seeing here on the x-axis is original 16S molecules from the beginning of the experiment. So these are essentially the barcodes. So this is specifically an experiment where we sequenced approximately 15,000 barcodes or 15,000 full-length 16S molecules. And then on the y-axis, what you see is essentially how many short reads were associated with each one of the barcodes at the end in the actual sequencing data. We call this the PCR bias plot, and people rarely look at this because they assume that they generally just ignore biases and PCR amplification. Since our technology is UMI-based, this is part of our automated pipeline's output, and we see this for every single PCR. And this is not a particularly bad PCR plot. So every PCR plot that we look at essentially looks like this. So what you can see is that molecules or barcodes from this part of the plot would seem like they were 10 times more abundant than molecules from this part of the plot if you were just judging by the number of short reads that originate from each one of these single molecules. But we know the ground truth, and ground truth is that all these 15,000 molecules were all single molecules at the beginning of the experiment. So this is just goes to show you that if you're not using UMI-based technology for correcting for your PCR bias in your long reads, you're going to get biased results in terms of composition. I think that Nick showed that this is also important for short read sequencing. If you're not showing that the LoopSeq or V1 to V9 was more accurate in terms of relative abundance compared to V3, V4. So that's for short read sequencing. For long read sequencing, it's even more important because the bias in amplification is even worse. As I mentioned, this is not this is a typical PCR bias plot. So I think it's safe to say that every PCR we've ever done looks like this. And this is probably the case for all of you that are doing PCR reactions. Some molecules get amplified very fast and some get amplified very slow. So that's just, I guess, the final point that I wanted to make regarding the advantages of loop long read sequencing compared to other long read sequencing methods.
Has GenomeWeb explored the possibility of applying your technology to enable single-cell sequencing?
Yeah, that's a very good question. So the answer is yes, we have explored this. So I should mention that we are not a single-cell technology per se. So there's nothing about our technology that enables you to specifically tag single cells. But our technology can be coupled to single-cell sequencing to enable single-cell long-read sequencing. So generally speaking, the field of single-cell sequencing has largely focused on short-read sequencing. So for example, if you look at, for example, products from 10x Genomics and other companies where they're doing a single-cell RNA-seq, typically what they're doing is short-read RNA sequencing focused on sequencing just one end of the transcript. But as we all know, in gene expression, arguably the largest source of diversity is isoforms and splicing. In the human genome, every gene has on average 10 different splice variants. So not looking at that and just sequencing the end of the transcript really leaves us blind to a lot of the variation in gene expression. So what I guess Loop Genomics' contribution to single-cell sequencing is bringing the ability to do long-read sequencing for single cells.
Okay, great. And does the ability to resolve closely related 16S sequences depend on the stringency with which you reassemble the LoopSeq reads? So
our assembly of the LoopSeq reads is completely independent of the downstream classification. Whether you're doing reference-based classification or OTU analysis, the de novo assembly, the assembly of the long reads is done completely de novo and is reference-free. So we assemble the long reads. We don't need, there's nothing about our de novo assembly of the long reads that is 16S specific. The pipeline for assembling 16S molecules in terms of the de novo assembly is identical to assembling RNA molecules or genomes. We don't need to know what the sequence is. It's completely de novo. And I guess maybe Nick, maybe you have something to add about that, but we're generating FASTQ files, and then the downstream classification is done completely independently.
Yeah, Nick, if you have anything to add, please chime in.
Yeah, no, I think that basically covers it because the assembly is de novo based on the barcodes. There's no stringency in that consensus sequence that Loop produces. When we get the FASTQ file, obviously, there are a bunch of benefits to the data being very accurate in terms of the types of stringency that you can use for like an alignment-based approach to a 16F database. And, in contrast, you have to make a whole different set of trade-offs with something like nanopore data. But yeah, no kind of specific stringencies upstream of the FASTQ file that come into play here.
Nick, do you have any suggestions as to how to minimize the false positives on the V3 and V4 analysis?
Yeah, that's a good question. So there are a bunch of different ways of doing this. Kind of informatically, and we actually do a bunch of false positive reduction informatically. So we'll eliminate what we think are spurious near neighbor sequences that are mismapping. But I think fundamentally the way to think about this is if you look at short read V3, V4 sequencing data, and you look at each read independently, it's a really hard problem to get rid of the false positives, right? So all of the different methods for reducing, and that's because of the fact that those errors look like real variants, or there is a sufficient amount of error that you mismap to the wrong reference. So at the individual read level, there's not much to do, and we're kind of in the territory of aligner design in thinking about that. But at the sample composition level, we do a bunch, as I said, kind of looking at near neighbors, and do we think that there's mismapping happening because of we're observing a little bit of kind of off-target mapping at lower identity to Bacteroides A, when we really believe that there's a lot of evidence for Bacteroides B in the sample, and we do that through a reference-based approach. You can also do that with a clustering-based approach. I think the data that we have shows that a lot of the kind of clustering approaches are fairly problematic in terms of also generating tons and tons of false positives. I think I would lean towards some of the newer methods, like a data 2, if you were going down the clustering route, and then classify the resulting kind of exact sequence variants that a tool like that produces. But again, that has a bunch of statistical assumptions about the data and parameterizes differently across Illumina, well, even across different Illumina platforms and preps and things like that. So I don't think there's a one-size-fits-all solution, unfortunately.
Nick, we have some questions about the process that you used for the analysis that you presented. One question is, what was the depth of coverage that you had for each approach that you talked about? We'd also have a question about the turnaround time for LoopSeq. And finally, one of our attendees would like to know which sample collection kits that you validated, and if you have any suggestions for sample collection to use.
Yeah, so I'm just writing those down. So on the sample collection side, in the scope of this webinar, there wasn't any, right, because these input standards were GDNA. So, nothing in the context of these known communities. We've certainly done work with others around sample collection devices and looking at, I think there's some good studies. There are a couple studies, I don't know if they're actually out of the Huttenauer lab, but that Curtis Huttenauer is on, amongst others. I'm totally forgetting the name of this consortium, but there's a microbiome sequencing quality consortium with a long name that I could probably Google right after this, that's done some work around producing kind of a homogenized, stool-like substance, and being able to put that in collection devices and kind of understand collection and storage implications. So I would look at some of that literature on the collection question. In terms of the LoopSeq turnaround time, once we get the data, it's very fast. I mean, the demo that I showed earlier, that was live, right? So yeah, I think the larger of those files, which is small still, it's like 50 megabytes compressed or something like that. That's probably a normal quote unquote file. Tuval could confirm, but this is the file they gave us. So I think it's kind of prototypical, and that takes 2, 3, 4, 5 minutes to process. Obviously, it scales with the depth of sequencing and the number of synthetic long reads in that sample, but minutes is the quick answer there. And then on depth of coverage, I'd have to look back at the data. It was very significant on the Zymo mix and on the major organisms in the ATCC mix. Obviously, we detect everything in all three cases with shotgun short read, with 16S short read, and with the 16S synthetic long read. So it's kind of sufficient in all cases. But I think the coverage varied from low single digits for those minority community members in the ATCC standard to a couple logs above that.
All right. Thank you very much. I'm afraid that's all the time we have for today. We'd like to thank Nick Greenfield and Tuval Ben-Yehezkel and our sponsor, Loop Genomics. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.